for our final panel, we're going to focus even one step further on a really extraordinary, hopeful event in the history of the AIDS pandemic. A little baby in Mississippi who made a lot of news. And with us is Dr. Deborah Persaud of Johns Hopkins University to help sort out the promise from the hype here. She was involved in the lab work to document the viral remission. And so let me just throw out at the beginning here about what you saw, being so involved in this field for so long, about what you first felt and thought when you saw the research here, the case here.
Sure. So before I start, I do want to thank EGPAF for supporting my research agenda and the National Institutes of Health. I think we would not have uncovered the Mississippi child without a 15-year investment in studying viral reservoirs in pediatric populations. Studying viral reservoirs in pediatric populations is a huge challenge, but is possible and is highlighted by the Mississippi case. So to answer your question, what did it feel like to uncover this case? I think it was really eureka. We found our N of 1. Prior to that, the field of HIV cure research was really catalyzed by the Berlin patient. Yes, it's a very intensive approach to cure, but it really provided proof of concept to the notion that actually HIV could be curable. And I think we can say today that the Berlin patient is indeed cured of HIV. And why can we say that? This is a patient who, and I think it's important to have definitions in place here. And that was actually our first mission when the Impact HIV Cure Scientific Committee was formed. We felt we needed to have a definition to define cure in clinical trial setting. And the adult field, so there was no definition really for pediatric cure when we stumbled upon the Mississippi case. So we borrowed the language from the adult field, and the International AIDS Society with Francois Boré-Senoussi and Stephen Deeks had published on these two broad categories of cure. One is sterilizing cure, and the other is functional cure. And with sterilizing cure, it means that a patient is known to have HIV infection, has established reservoirs, and you need to actually completely clear those reservoirs for the individual to be able to go off antiretroviral treatment and stay off antiretroviral treatment without having virus rebound. So in other words, virologic suppression off antiretroviral treatment. With functional cure, an individual can still have HIV-infected cells, but it's actually the immune response that controls the virus from replicating to the point where there's rebound viremia. Now, with respect to the Mississippi child, the child was neither, we didn't know was sterilizing cure or not, but we used the word functional cure because that was really the only definition available in 2013. It certainly makes people pause. What does it mean to be functionally cured? We felt it would be scientifically negligent in such a short term of follow-up. At the time, the infant had been off treatment for 8 months. When we first studied the infant, we felt it would be scientifically negligent to not have pause when we reported the case. So it was indeed, we have our N of 1. We need more than 1. We need long-term follow-up. But really, this case solidified the biological basis for a cure agenda in pediatrics. And it was a setting in which we were really prepared. To quote Louis Pasteur, we were really, this was not by chance. We had prepared minds in handling these cases. We had, in response to the NIH initiative research priority areas, the IMPACT Network, or the International Maternal Pediatric AIDS Adolescent Clinical Trials Network, had identified pediatric cure as a priority for the network. So in November 2011, I was elected to lead the cure committee for the IMPACT Network. And it's in that framework that we began development of cure strategies for pediatric HIV/AIDS. And the first strategy and the lowest hanging fruit for us was really very early treatment of neonates. So we had started to develop the P1115 protocol that's now opened, but with the context that the drugs we have available for treatment was really inadequate to achieve cure. we thought we needed more than AZT, 3TC, and nevirapine, which is what we have available. Those drugs are used for prophylaxis. We felt we needed an integrase inhibitor, an agent that blocks the virus from integrating into the host genome. And so to pave the pathway towards a cure clinical trials, we actually started in collaboration with Merck a pharmacokinetic study, and those studies are ongoing, of raltegravir. P1097 is looking at the washout PK, and Lynn Moffason is part of that group, washout PK of raltegravir in infants born to women who received integrase inhibitors. And that's completed, and the second protocol was P1110 to get the dosing information. How do we multi-dose raltegravir in HIV-exposed infants to use as very early treatment for cure? We had in place, okay, how are we going to define cure? The way we define cure for pediatrics is being really a state of our remission, where an infant would receive very early treatment, would be able to stop treatment and have no experience of virus rebound within 48 weeks of treatment. So that was our working definition of virus remission in the context of very early treatment and cure. And then came the Mississippi Child, and I think the Mississippi Child, as Doug mentioned, really represented collaboration and investment in the field. And there's been significant investment in HIV cure research for adults, with formation of large collaboratories, of partnerships between industry, scientific investigators, and NIH partnership for adults. But no such collaboratory existed for pediatrics. And to borrow from Shafiq, we hitched on the adult bandwagon and tried really hard to engage those investigators in our research agenda. And so in encountering this case, I think it was really a case of a pediatrician who had started a child in Mississippi. And again, this highlights that we have a problem here in the United States. It's not about international. I mean, the CDC has reported there are over 200 cases every year in the United States. I think in 2010, the year the Mississippi Child was born, there were 210 new infected cases in the United States. And in fact, if you look at the map, the Mississippi Child is not on the map of the 210 cases, which means we have gaps in identifying these kids, and there needs to be more intense attention to surveillance within the United States.
Can I ask you, just to interrupt, because you made an interesting point that I haven't seen very much in discussion of this. Sometimes it's viewed as a bolt from the blue, but there was actually a research structure in place to recognize that this was a big deal and then be able to act on it rather quickly. If it had come much earlier, that might not have existed.
I think we would have missed it. I think we would have missed it.
Right. But it's an interesting point about the way science progresses, is that you have that structure, you're prepared for breakthroughs, right?
Yes.
I mean, is that fair?
I would say that. And I'd say the pediatric reservoir study agenda started on the basic principle. When we started in 1998, when Bob Selcano and Tony Fauci's group and Doug Richman's group really provided the sobering news that HIV is incurable. When I entered into the field under the mentorship of Bob Selcano in 1998, the fundamental basis for the pediatric studies was that the immune system is different in children and adults. It's a developing immune system. Memory T cell formation is slowed. And perhaps the dynamics of this reservoir will be different in children and adults. And so all of the tools, and actually to put this in perspective, studies of our reservoirs in adults require a single blood draw of 180 to 250 mLs of blood. That's a total blood volume of an infant. So we first had to establish, I think people don't realize that we first had to establish methods in place. Is it really feasible? And I don't think anyone would have embarked on this field without knowledge that actually the children have higher CD4 positive T cells. You have per mils of blood, you can get a lot more cells to study. And we've scaled those studies down in the Impact P1030 trial of early treatment in children. We've been able to study reservoirs on 3 mils of blood. Now, that requires overnight shipment and fresh processing, but certainly the agenda for reservoirs was initiated on the basis of the distinct differences in the adult immune system and the pediatric immune system. And the Mississippi child highlights that, but again, it's an N of 1. The Berlin patient is still an N of 1. And so we need to be able to replicate that case to say for sure that this is really not some other factor that has contributed to the outcome in this child.
Tell me a little bit of the story about how long it took to confirm that the child was actually cured, but also then the decision to announce it very quickly. I'm just curious how that worked.
So I think I'll write a book in 5 years on the Mississippi child, but to just sort of set the stage, it's the way the case was identified is Dr. Hannah Gay was actually part of the Pediatric AIDS Clinical Trials group. She was a clinical trials, head of the clinical trials site in Mississippi, and participated in the first early treatment trial of pediatric HIV in the US, the PSATG 356 trial. And in that trial, she worked with Dr. Catherine Zuriaga, who was the principal, the protocol chair for that trial, and the immunologist involved in reporting of the Mississippi case. And it was through that collaboration or a relationship that existed. Whenever Dr. Hannah Gay had a difficult case, she would contact Catherine for advice on how to manage these kids. So here she was, knowing that this child, she had diagnosed this child as infected, had started the child on treatment, and now at 23 months of age, the child no longer has evidence of antiretroviral treatment of HIV infection. And so it's that, and that's how I got involved in this case. So what did it take? I'd never met Dr. Hannah Gay before. So my lab, as we do, we get de-identified samples and we just process them. Is there a latent reservoir that exists? If it does, how much? And we have multiple assays that we can look for, but the critical assays, is there replication competent virus still present in the memory CD4 T cell in this child? And so the first sample, we cultured over 20 million replicates of cells, and there were no virus detected in any of the cultured wells. And we just don't see that when someone's off antiretroviral treatment in all of the culture assays we've done over 15 years in pediatrics. So I felt fairly confident that at least at that time point, there was no detectable replication competent virus. But of course, we had to convince ourselves this child was indeed infected. And I think this is the challenge for the field, is that we can start kids very early on one test or a point-of-care test, but there can be false positives, there can be mislabeling and sample mix-ups, and it's highly critical for the clinical trials that we have multiple samples collected to really convince ourselves that we had infection in a child that was then cleared by the regimen. And so it actually took a lot of detective work to convince myself first before, this is my scientific career, before I go out on a limb and stand in front of an audience of peers to say this child is likely, I didn't say cured, is likely, has achieved virologic control after starting antiretroviral treatment. And so in fact, if Dr. Hannah Gay had not carefully documented serial viral load measurements in this child over the first 3 weeks of life, we would not have been able, and we still have not been able to convince everyone that this child was indeed infected. So we had four independent blood tests over the first 19 days of life documenting HIV viral nucleic acid or RNA in the plasma of this child with the classic decay in plasma virus that we see over the time course of treatment as expected based on turnover of cells. And the child had DNA positivity. So I think I was able to convince myself this was clearly a kid who had met our standard definition of infection and responded to therapy as we would have expected, but did not rebound off antiretroviral treatment. Now, in terms of the way the story unraveled, so we first got called in September of 2013, in 2012 about this child. And in December 2012, the NIH was having their internal meeting with a group of experts to update on the Martin Delaney collaboratories on HIV cure. And they invited me to speak on a pediatric cure agenda as a representative of the Impact Network. Now, there was no way I could stand up at a podium at an HIV cure symposium at the NIH, I felt, and not disclose this case. It had been 3 months. We'd had 2 samples. both samples showing the same thing. The child had not been restarted on treatment. The question for us was from Dr. Hanegay was, should I start the child on treatment? Because she knew the kid was infected, and we felt 5 months out with no rebound is so atypical of perinatal HIV infection. Most of these kids rebound 2 to 4 weeks, that we felt it was safe with close monitoring to follow this child. And so at that meeting, I presented the preliminary data with just not as much detail as we have today, but I felt it was important to disclose to the scientific community our findings. It was received with optimism, but a lot of skepticism. And then we decided that it was scientifically responsible to, given that there was a cure session at CROI devoted to cure in adults with no pediatric representation on the platform, that it was important for us to keep the agenda in the spotlight, and decided to submit it in January as a late breaker for CROI. Now, I think the bar for acceptance for late breakers at CROI is very, very high. We felt that acceptance in that meeting meant we had sufficient laboratory data and information to support indeed that this was a case of remission. And it was accepted and presented. I will say I was very naive going into that meeting in terms of the media hype and hope around this case. And there certainly was a lot of hype. We didn't intend that was not the intent. It was just sharing of scientific findings, and we hoped we did it in a responsible way, and ultimately was published. I think it does provide hope, but it provides a lot of hype. That there needs to be a balance, and I'm glad to say that the clinical trials, with that we were actually really able to develop quite quickly a clinical trial that we now have open around the world to be able to replicate the Mississippi trial.
2 questions before we just take a couple of questions. One of them, just stepping back a little bit, you have a compelling case that convinced you, but just for a non-scientist, describe the biological model of why, plausible explanation for why the case exists.
Sure.
I'm curious.
I'll say, but before I say that, I think one important comment that was made in terms of was this a case of switched infant syndrome? And this does happen in nurseries where kids are switched. And so we did do HLA typing to confirm that indeed the mother and child were linked. But to get back to your question
about the model, the biological.
So the biological model is simple, and that is when a newborn, the amount of memory T cells are negligible. It's less than 5%, somewhere in the order of 5%, measured in the peripheral blood. And so your memory T, so one major reservoir for HIV, that's the barrier to cure, is this reservoir for HIV in memory CD4 positive T cells. Within memory T cells, these cells are very quiescent or latent. The virus can persist in a latent form. So because those cells are quiescent, there's no virus expression. So those cells are invisible to the immune system. They're invisible to the current antiretroviral drugs that we have. And in fact, a major strategy for adults is actually to try to reawaken those latent or sleeping genomes to allow the virus to be expressed so those cells can be killed. So in a neonate, there's actually very slow, it takes actually through up to 15 to 20 years for your memory T cell compartment to develop. And it's very slow in the first few years of life. The neonatal immune system is also tolerogenic. It's slow to react because it's so primed to not respond to maternal antigens, because that would be a bad thing in utero to respond to your mother's antigens. And we believe that it is this immature, or not immature, non-developed immune system that promoted, or that can promote very early treatment and blocking of virus reservoirs from forming. That's the hypothesis. We haven't proven that yet.
But it's plausible.
It's plausible. It is plausible.
And then just to follow up about the status of the research efforts right now that are coming out of this. Just tell me where we are and where we're headed.
So the Impact HIV Cure agenda is twofold. One is, of course, focusing on very early treatment to block reservoirs from forming, but then we have most of our infected kids are chronically infected, as Doug mentioned, and so immunotherapeutics is a major strategy for this population, and so we have capsules planned for development looking at the VRC01 monoclonal antibody to see if these can reduce viral reservoirs in chronically infected children. There's a plan to study vaccines, autologous virus vaccines with Argos Therapeutics, in collaboration with Argos Therapeutics, to look at T-cell-based vaccine approaches to purge viral reservoirs. Certainly there are strategies to modify CD4 positive T cells so you can render them CCR5 delta 32, heterozygote or homozygote using zinc finger nucleases. So we have a robust 5-year agenda, plans that we work synergistically with the ACTG group to be able to move strategies very quickly into our pediatric population, obviously, starting with those who are 18 years of age. Right. And older. So we're very excited about a pediatric cure agenda. But to get to your point in terms of the neonate, the unique aspects of perinatal HIV infection is we know when infants are exposed, the timing of infection is known. So any of these strategies, immunotherapeutics combined with very early treatment, I think shows great promise. And to build on the model that Lynn mentioned earlier is, the pediatric model is the mother-to-child transmission model is what set the stage for the use of antiretroviral drugs to prevent transmission. That's translated into prevention strategies for adults. We know now there's a single adult case that has been treated very early, in which the findings appear very similar to the case in Long Beach that we just reported. So I think there's converging evidence here that it may not just be for pediatric infection. It's just there may be a window of opportunity to intervene with blocking virus reservoirs in acute infection if you could identify it.
Well, let me take a couple of questions if we could here. I don't know if any of our panelists might even have a question, but please, maybe Gwen. All right. Why don't you wait for this so others can hear?
On the Mississippi baby, how was it that baby was so closely monitored with serial viral loads over 3 weeks? Was she part of a clinical study or, that's not the norm?
That's a very good question. And what happened is, so the child was hospitalized, was slightly preterm. So was hospitalized in the first few weeks. So it was because of being in the hospital, sampling for viral load testing was feasible. And it was done because the pediatrician was concerned. This was a mom who was not engaged in care. She didn't know she had drug-resistant virus or not, and initially started a nevirapine-based regimen. She was concerned about pre-existing resistance, and so monitored viral load closely to assure that there was response to the drug regimen that she prescribed. And it wasn't until the diagnosis was confirmed that she switched the child over to Kaletra-based regimen. And the reason she switched to Kaletra, this is all the pediatrician's work. This has nothing to do with me or Dr. Luzuriaga. The rationale for treatment, the follow-up, and the reason she switched is there were social issues, which is the major issues in any of these cure agendas or treatment agendas. These families are fragmented. There were social issues, and she felt by switching to Kaletra, even though the child was responding to treatment, it would actually, there would be a lower barrier to resistance, a higher barrier to resistance. So she was very concerned about non-compliance and resistance and switched to a Kaletra-based regimen. So it was the frequent, without that, I don't think we would have been able to convince the scientific community that this kid was indeed infected.
Maybe here, and then we'll do Doug.
Thank you. I'm Regan from UNAIDS, and thank you for your work. This is fascinating. Is there a relationship potentially between the ability to limit the viral reservoir and priming a child's body for a therapeutic vaccine down the line?
Yeah, thank you for that question. I think if we had a vaccine product, that would really be the ideal setting, because to rely on antiretroviral drugs solely is problematic because of issues with adherence, issues with bioavailability, illnesses that could compromise constant intake of drugs. So I think certainly to model on any infectious disease system, an effective immune response is the best strategy in any persistent viral infection, varicella, herpes, EBV, it's an effective CTL response or humoral response that controls virus in those settings. So I think really, if we had any vaccine product, that would be the best next step, is to look at combining antiretroviral drugs with an immune-based therapy. And certainly with VRC01, you know, the studies are, there's a protocol written for VRC01 for prevention of mother-to-child transmission. We hope with our studies of VRC01 in the older chronically infected children that we can begin, as safety data are collected in the pediatric population, that we can begin to move the product down in combination with antiretroviral treatment. So, and if there's some T-cell based vaccine, of course, that would be a great opportunity to study that effect. I will point out that with very early treatment, we actually preserve immune function, and we've identified now a cohort of kids in the US who've been treated now since 2 to 3 months of age for 15 years. And in fact, to our surprise, the dynamics of the reservoir is very different in those kids. They actually have decay of the reservoir, which is not described in adults. And Catherine and I have a paper that's now coming out in JID showing that there's actual decay of proviral reservoirs in these kids over 10 to 15 years. So lots more to learn in the pathogenesis of viral reservoirs in these children, but much more work to do in terms of the central nervous system and the gut, which raises the issues in terms of ethics. And there's a beautiful paper by Seema Shah that's just written, published in Lancet ID. Christine Grady is the senior author. I'm a co-author on that, and really the need for ethical foresight in cure-related studies. And you know, the issue of treatment cessation in kids who are responding well to therapy is a big one. And it represents a gap in knowledge. We currently don't know how to identify a kid who's been cured unless they go off therapy. And we're going to have to do those studies. But as Lynn said, we're doing those in a very cautious way with frequent viral load monitoring. Treatment cessation has been part of clinical trials, the SHARE trial. In South Africa, it's been done well. I think it's a great example of how these studies can be conducted in resource-constrained settings very beautifully, and we will model our studies based on that.
Doug, do you want to? Do we have a mic? Okay.
Do you want to go to Doug? Or
No, we'll do. You can go ahead, and then we'll do Doug last. It'd be great.
Thank
So
you, and
we'll
thanks
take-
for a great panel. Trish Carlin with EGPAF. I just want to follow up on the window of opportunity
Yeah.
point that you made. I'm curious if any of the research studies that are planned will talk about how early is early enough
Yeah.
in terms of achieving suppression and functional
Yeah,
cure.
that's
Thanks.
a great question, and we don't know that just yet. When we first built P1115, we thought we would look up to 7 days in the animal models. In Ashi Haas's group, 7 to 10 days was the window of opportunity. But certainly, with the need to kind of get this trial out on the ground to quickly enroll, we decided to focus on the first 48 hours of life. We hope to be able to build on that to look at the window of opportunity beyond 48 hours of life. Studies by Dan Baruch actually recently showed that 3 days, I think, may be too late in the animal models, but this is where we can be aided by animal model systems. Hopefully we can build on these trials as we go forward. Many, but the Mississippi child has changed delivery of pediatric care and treatment. I think it's, to echo what Shafiq said, even if we don't achieve a cure, we'll achieve through this mission very early treatment, retention in care, and diagnosis in children. But I'm very hopeful that we'll achieve replicates of the Mississippi child.
If it's the last question, I'd also like to give my thanks to Debbie, Catherine, Hannah, and the fantastic scientific work that they have done. And as a scientific peer, I congratulate you. My question is also scientific, and since I was a premature baby, changed a bit now, I also was particularly interested in that element of this case, and wondered, as the fetal-adult layering hypothesis is developing steam, whether you think that this actually may be related to the premature stage of the child, and the fact that there may have been a different complement, and so the generalizability may reflect identifying individuals who have a greater fetal-adult layering.
Yeah.
And sorry for the technical jargon.
Yeah, no, I
there.
think that's a great question, and actually we were funded, or RO1 was funded by NICHD, and a component of that is looking at the, Mike McCune's model of fetal adult layering, and Mike is, we're collaborating with Mike on that and providing samples in the context of PMTCT programs to look at that question. And that is intriguing, is the extent to which prematurity contributed to the outcome in this child. And even if it did, a substantial proportion of HIV-infected infants are actually premature. They're not term infants. And last year at CROI, I also felt the need to disclose the second case, because on the heels of the Mississippi child, many pediatricians or care providers have started to, and in fact, there are probably about 4 kids around the U.S. who've now been started very early on treatment in the first few weeks to months. So again, highlighting, we have an epidemic here in the U.S. in terms of new infections. But the Long Beach child was not premature. Now, obviously, that child is still on treatment, but certainly with very early treatment, it's become very difficult to detect traces of HIV infection in that child. But it's probably multifactorial, fetal adult layering, very early treatment, tolerogenic nature of the neonatal immune system, which together leads to an outcome, a favorable outcome in this setting.
Let me use the moderator's privilege just to maybe ask one more question, because I want to know, what are some of the reasons from your involvement in this whole process of why HIV services aren't reaching women in the United States? What problems do we still have here?
Let me ask Emily to comment on that. I mean, it's really failure to test frequently. Right now I think we recommend testing in the first part of pregnancy, but not towards the end of pregnancy. We certainly have mothers seroconvert during pregnancy. Often they're negative, and so testing the infant can be negative because they haven't developed an antibody response. There's fragmentation of social services, attention and retention in care. Many of these women have other children, so somehow they fall through the cracks. Again, they're not engaged in care. So we need to have some active surveillance system focused on identifying women at risk, frequent testing of women at risk, frequent testing during pregnancy, and really engagement and retention in care for the family's health.
Well, let me just conclude by saying, just in general on these panels, how impressed I was. There was significant agreement on making this a central focus within a broader effort to reach mothers and children as being central in the United States and abroad. We seem to be on the verge of some extraordinary possibilities when it comes to diagnosis and treatment and even functional cure. And I'm always just impressed to meet some of the scientific heroes of our time, people that are making extraordinary difference. I think historians will look back on this period and look at the people that were involved in vaccine research and AIDS research as some of the great scientific heroes. And we're fortunate to be a part of that. And thank you so much for being here.
Thank you for having me.